is about transcription, which is the process of how RNA is assembled and built based on the DNA sequence. mRNA, please take my instructions to the ribosomes.
I got Captain. Oh, powered. Ready?
Transcription. A transcription unit is a sequence of DNA that will code for a polypeptide to be built, or RNA, transfer RNA, or ribosomal RNA. The enzyme that is important for transcription is RNA polymerase. What RNA polymerase does is it separates the DNA strands from one another and will actually do the transcription of mRNA. When you see the word transcribe, what that means is the appropriate nitrogenous bases or base pairing off of the template strand of DNA, the matching up. Again, the A's go with U's and the C's go with G's. mRNA is made from a 5' to 3' direction, so that's the way that RNA polymerase builds. And as a reminder, uracil is going to replace thymine. Again, there is no T in mRNA or any RNA strand. Now, what RNA polymerase does is it attaches to a promoter. A promoter is a specific gene sequence where this enzyme is able to attach. At the end of the gene, it is going to have a code that is considered a terminator where RNA polymerase knows to detach. The promoter will be at the start of the gene, so it'll be considered to be upstream, and then the terminator is going to be towards the end, which is known as being downstream. There are going to be 3 main steps for transcription. The first step is initiation, and in prokaryotic cells, RNA polymerase binds directly to the promoter, the piece of DNA that is going to start transcription. In eukaryotic cells, initiation is a little bit more complex. A DNA sequence is known as a Tata box. Now, what exactly is a Tata box? A Tata box has a sequence of TA, TA, A. This is going to be on the non-template strand. So the strand that is not being transcribed. It is in the promoter region of the DNA. So the promoter region is going to have this sequence. It's also going to have that AUG start mRNA codon. Now, transcription factors, this is going to be a location where different enzymes in proteins attach to the DNA that needs to get transcribed, and RNA polymerase knows where to attach. It is also going to include the Tata box as well. The second step of transcription is called elongation. This is actually the process of RNA polymerase adding in the appropriate nucleotides or nitrogenous bases to that template DNA strand. Remember, when you're doing transcription, you need to work off the 3' to 5' end of DNA, and you are building an mRNA strand in the 5' to 3' direction. The way that you work is from left to right, and again, left to right is known as working downstream. In addition to RNA polymerase adding in the appropriate nitrogenous bases and nucleotides, it also is responsible for the untwisting of DNA as, again, it is being transcribed, and it is also responsible for rewinding it back after the mRNA strand is made. If you look at the picture, this summarizes the first two steps in transcription. At the very top, you can see the promoter region, which has the big blob, which is RNA polymerase attached. You can also see that there's a start point. Now, what's not being shown is upstream to the left would be that TATA box. Even though it isn't being shown in this picture, it is there. And then initiation takes place. RNA is going to be transcribed or built by base pairing rules, and then the elongation is where RNA polymerase works downstream to add in the appropriate base pairing letters or nucleotides. The third and final step of transcription is known as termination. So what ends up happening in termination in a prokaryotic cell or a bacteria cell, RNA polymerase is going to transcribe or code a terminator sequence. Once that terminator sequence is made, RNA polymerase is going to stop making RNA and detach itself downstream from the mRNA strand. Now, it's a little bit more complex in a eukaryotic cell. RNA polymerase is going to make a polyadenylation signal. Now, what that is it is a code. The code of this signal is AAU, AAA. Once mRNA has that 6-letter code, similar, the RNA polymerase is going to detach itself, and downstream the mRNA will be cut. Now, again, similar, but at this point, the RNA strand that is made in a eukaryotic cell is called pre-mRNA, because there's a little bit more fine-tuning that needs to take place in a eukaryotic cell before that mRNA escapes the nucleus for polypeptide or protein synthesis. In a prokaryotic cell, once there's that terminator sequence, the mRNA is actually ready for the second step known as translation, which will be the building of the protein.